Hello, this is Daniel, and today we'll discuss about the senses and how we perceive them and how those are related to our brain. Let's get started. So let's begin with the vision. You may have heard this information that your right brain processes the information from the left side of your vision, and your right and your left brain process the information from the right side of your vision. So basically, this model shows that in terms of the right eye, the objects in the left side of your vision, colored in this red part, is reflected to this side of your eye, and it moves along this nerve tracts, and ends up in the right first visual cortex in the occipital lobe. And this right side information is reflected to the part of the eye that is closer to your nose, and this crosses the brain and then is stored in the left visual cortex. Same thing happens in the left eye. The left part of the vision is reflected in the part of your left eye closer to the nose, and then it crosses the brain from left to right, and then it ends up in the right side. And this part of your left eye reflects the information about the right side, and it goes along this track to end up in the left side. So basically, the information on the right side of your vision is processed by your left brain, and the objects in the left side of your vision are processed from in your right brain. So diving technically, on their way to the brain, signals travel along nerve fibers from both eyes, which converge at a part called optic chiasm, this X-shaped part, which contributes to transporting the left information to the right brain and right side information to the left brain. Then the information proceeds to the either side of the brain based on their first perception point, and then then the visual information is relayed to lateral geniculate nucleus, LGN, labeled here. So they cross or just go along their neural tract at optic chiasm, and then they reach LGN, lateral geniculate nucleus, which is a region of thalamus, and thalamus is a brain part that manages most of the sensory information except smell. And then they reach the primary visual cortex at the occipital lobe. This is basically how vision works, but in a very brief manner. Next is hearing. So hearing involves a series of steps to convert sound into electrical signals that can be carried to the brain and then be perceived. Sound in the form of air pressure reaches the pinnacle of your ears, which is this part. And then the waves are inputted to the ear canal to reach the eardrum, or in other words, tympanic membrane, so this part. This tympanic membrane vibrates in response to these changes in air pressure perceived through this outer part of the ear. And these vibrations are sent along the three sound amplifying bones, malleus, incus, and stapes. The last bone in the chain, the stapes, acts like a tiny piston, pushing the oval window, a membrane that separates the air-filled middle ear from the fluid-filled cochlea of the inner ear, which is this one. The oval window converts the mechanical vibrations of the stapes into pressure waves in the fluid of the cochlea, which they are transduced into electrical signals by specialized receptor cells, which are also called hair cells. These hair cells are located on top of the vibrating basilar membrane, which are the membranes inside cochlea. And when the membrane moves up and down, the hair-like cell bends against the overlying structure called tectorial membrane, which opens small ion channels in the hair cell that allows ions in the surrounding fluid to rush in, which converts the physical movement into electrochemical signal. Then the hair cells are stimulated in this way and excite the auditory nerve, which then sends the electrical signals onto the brainstem and thalamus, and then which are sent to the auditory part of the cerebral cortex. Then let's move on to taste. So our ability to taste foods depends on the molecules that are set free when we chew or drink. These molecules are detected by taste cells with taste buds located on the tongue, which are shown in the red color in this photo, which are along the roof and the back of the mouth. You start to lose those taste buds around the age of 50, and each taste buds consists of 50 to 100 sensory cells, which are receptive to one of the 5 basic taste qualities, which are sweet, sour, salty, bitter, and umami, which is savory. Now, contrary to common belief, all tastes can be detected across all parts of the tongue. So when the taste receptor cells are stimulated, they send signals through 3 cranial nerves: facial, glossopharyngeal, and vagus nerves to taste regions in the brainstem. Then the impulses are then routed through the thalamus to gustatory cortex in the frontal lobe and insula, where specific taste perceptions can be identified. Next one is the smell. So initially the smell odors enter the nose on air currents and bind to specialized olfactory cells on a smell patch of mucous membrane. Axons of these sensory neurons enter the 2 olfactory bulbs after crossing through tiny holes in the skull, shown here. From there, the information travels to olfactory cortex, and therefore the smell is the only sense that does not go through thalamus before reaching the cortex. We have around 1,000 different types of olfactory cells, but can identify about 20 times as many smells. And the tips of olfactory cells are equipped with several hair-like cilia. They are receptive to a number of different odor molecules, and many cells respond to the same molecules. Specific smell will therefore stimulate a unique combination of olfactory cells and create a distinct activity pattern. Olfactory information can also pass to nearby brain areas where odor and taste information are mixed, and which creates the perception of flavor. Now, size of the olfactory bulbs and the way neurons are organized can change over time. So the last part is the touching perception. There are several types of tiny receptors embedded under the dermal layers of the skin. In this model, there are Meissner's corpuscle, Merkel's disc, Ruffini end organ, sweat gland, Pacinian corpuscle. So Merkel's disc and Ruffini endings sense different aspects of touch. And all these receptors have ion channels that open in response to mechanical deformation, triggering action potentials that can be recorded experimentally by fine electrodes. And the first 2 types of receptor adapt quickly and so respond best to rapidly changing vibration and flutter. Merkel's disc responds well to a sustained indentation of the skin, which is pressure, and Ruffini endings respond to slowly changing indentations. The right image illustrates the distribution of the sensory neurons and receptors in our body, where the parts with more sensory neurons and receptors are drawn bigger. This is it for this video, and thank you for watching. See you in the next video. Bye!